3 points. Number one, the changing face of treatment, really going over a little bit of the same ground. When you look at where we've come from 1983 right through to 2018, and I put 2018, 2 drug regimens and long-acting drugs as being where I think the interest for immediate development is going to hit us. So that's where we've come from, and that's potentially where we're going. If we look at all the guidelines, 29 drugs have been approved. Now the guidelines, EACS, US, ISUSA, WHO, we're actually dealing with 11 drugs out of the 29 in our preferred regimens. And of those 11 drugs which are listed at the bottom, we're actually talking now about 3 of those drugs for 2 drug regimens. That is Dolutegevir, rilpivirine, and 3TC. Plus, from the new antiretrovirals, from phase 3, 2B3, we're talking about Cabo Tegravir as well. So from all the drugs that we've had and the drugs which are in development, we're currently talking about 4 drugs for 2 drug regimens. So let's just talk a little bit about these 2 drug regimens. What is the evidence from a pharmacological perspective that we might be onto something? So why should we consider 2 drug regimens at all? Well, why take 3 or 4 drugs when 2 can do? We can use that paradigm for preservation of future treatment options, potentially reduce the impact of long-term exposure to multiple antiretrovirals, fewer drugs, as Jonathan was saying, potential to reduce DDIs, although clearly that depends on which drugs we're talking about, taking out of a regimen, establishing a new paradigm which can take us forward. Will it have cost implications? Maybe a question mark against that. So what would make an ideal 2-drug regimen? Well, it's got to be a potent antiviral combination. It's got to have a favorable PK profile, maybe less DDIs. This high barrier to resistance, fewer short-term, long-term adverse events. And it's going to give us reduced cumulative drug exposure over time. If this is lifelong, then less drug exposure that the body is going to have. And again, can we make it more affordable? So is Dolutegevir a core drug? Because that seems to be at the present time a drug which is being in that 2-drug regimen. Is Dolutegevir a core agent to support 2-drug regimens? Well, if we look at all the literature and what we know about Dolutegevir, it's got a rapid antiviral activity. It's got a fairly high barrier to resistance from all the data. It's got this long binding to wild-type integrase, and that dissociation means that not only is it the PK half-life, but it's the binding of the drug to integrase, which clearly can be important as well. The DDI issue is one which I'm not going to go into in any great detail, and it's got this long half-life, again, which Jonathan showed, which is clearly important for us. And we've got a lot of clinical trial data on Dolutegevir. So Dolutegevir as a core agent to support 2DRs has a number of features which I think are favorable. So what about combination then? And we're talking about combination with either 3TC at the present time or rilpivirine in the 2-drug combination. So 3TC, well, we've had a lot of experience with 3TC. It's in all the major guidelines. I think you'd agree 3TC is well tolerated. It's got this long intracellular half-life, which is important rather than the plasma half-life. 3TC, hardly any DDIs with 3TC. And we've got clinical data emerging on 2-drug combinations. What about rilpivirine? Well, we've got some data emerging with rilpivirine in combination with another integrase, that is Cabo Tegravir as well. So we have some data from that, which is emerging. It's got a long half-life, important for us to consider. And the tolerability, certainly as an NNRTI, is potentially better than other NNRTIs. So there are a number of features which really point to the fact that we have drugs which can be useful in combination. So then what about Cabo Tegravir as another integrase in combination as well? And the combination, again, which we've mentioned, is Cabo Tegravir in combination with rilpivirine. So Cabo Tegravir is an integrase inhibitor. We've got an oral formulation. It's got a half-life of 40 hours. It's forgiving. It's got a long half-life. But then this nano suspension gives us a half-life of days, 20 to 40 days. And that in combination with rilpivirine, you can see the balanced half-lives of the oral formulation, 40 hours versus 50 hours, and also a rilpivirine nanosuspension, where you've got a long half-life, somewhere between 30 and 90 days. And we have the data from LATE 1 of proof of efficacy for the oral form, and we've now got the intramuscular form of Cabo Tegravir and rilpivirine, which has maintained viral load less than 50 in LATE 2. And just to summarize all the half-lives of these different drugs that we're talking about, because you've got to get some sort of balance. If you've got a 2-drug regimen, you need some balance in the half-lives. Dolutegevir, 14 hours, maybe a bit longer, as we'll see in a moment. The 3-TC triphosphate, around 22 hours, so that's balanced, the DTG 3-TC. Dolutegevir, rilpivirine. Rilpivirine's a bit longer than dolutegevir, but overall, they're both long half-life drugs. And then Cabo Tegravir and rilpivirine, the balance of the oral and the nanosuspension is pretty good. And note just at the bottom there that Bictegravir half-life is around 17 hours, so it's around the same as dolutegevir. So this is the long-term PK, and this was a study done at the unit in Chelsea in Westminster where Mark is, looking at a comparison of dolutegevir versus elvitegravir. So I think from a PK, this is why we're looking at dolutegevir, because the half-life is clearly very long, and you can see that in that upper line. And the point with the circle around at the 96 hours is around the protein-adjusted IC90. So from an oral perspective, you don't advocate it, but you can miss a dose and you're still above the IC90, the protein adjusted for wild type, certainly. Comparing that with elvitegravir and cobicistat. So once you stop or miss a dose, cobicistat begins to crash, and therefore the elvitegravir also begins to decline as well. So there is a difference in the exposure of the two integrase inhibitors. And just comparing, there's lots of parameters that pharmacologists talk about, and sometimes they confuse themselves. And you've got all these IC values. That's the information from in vitro studies, IC50, IC90, IC95, protein adjusted. We have that for all the integrase inhibitors. The EC, which is the in vivo parameter, the effective concentration, which affects 50% of your viral load, depression, for example. And then we have the pharmacological parameter. And from that, we can get IQ, the inhibitory quotient, C trough, the trough concentration over the inhibitory concentration. And again, dolutegevir is higher than elvitegravir and raltegravir, and Cabo Tegravir is probably higher than dolutegevir. So we've got this IC value compared to the trough concentration, which gives us an IQ against the wild-type virus. Now, that isn't telling us against mutant 1 or mutant 2 or whatever. That's telling us what it is against the wild-type. But if you look at this data on genetic barrier to resistance for specific antiretrovirals, and along the horizontal axis, the genetic barrier to resistance, the approximate number of mutations needed to fail versus the potency of the drug in terms of the change in viral load, you can see that dolutegevir here has got this IQ of 17 to 19, which is higher than elvitegravir, and this links then to the genetic barrier to resistance. But note that for the boosted PI, you've got an IQ of 76, and you've got a slightly higher genetic barrier to resistance as well. So comparing it to the PI is higher than the dolutegravir. But in combination with 3-TC or in combination with rilpivirine, then what we're looking for is a fully suppressive therapy. Because what you've got to have are concentrations of drug uniformly achieved at all the sites where the virus is replicating. And that's going to be determined by the drug. It's going to be determined by the drug penetration, not only into the PBMCs, but also into other sites where virus can be. And those factors can determine how much of the drug, how quickly the drug, the effectiveness of the drug to get into all the sites of viral replication. And so we've got to think about not only plasma and PBMCs, we've got to think about the CSF, we've got to think about the male and female genital tract. And so here, just comparing dolutegravir with darunavir, you can see how similar that the ratio of the CSF or semen or cervical vaginal fluid is to blood plasma, 0.02, 0.01 for Dolutegevir, 0.07, 0.17, 0.06, whatever, 0.09. So the ratios are pretty similar for Dolutegevir and for Dolutegevir as far as access into those sanctuary sites are concerned. Now, can we maybe save something in the patient? Well, just look at how much drug patients are taking. If you take a 3-drug regimen of Dolutegevir plus FDC TDF for a year, you will take 0.5 kilogram of drug. Okay? But come down to a 2-drug regimen and you take Dolutegevir plus 3TC, you'll take 1/4 of that. But Cabo Tegravir plus rilpivirine oral, you'll take 20 grams. And if you go down the route of an injectable, you will take 6 grams of drug in a year. So that's a bit of a difference from 0.5 kilogram of drug versus 6 grams of drug in terms of what the body is seeing. Now, what does it mean? Okay, maybe it's a slide which is visual, but then we have to determine what actually does that mean in practice. So finally, can we move to the long-acting? We've heard a little bit about long-acting. Can we move? Well, this is a review that was published just a week or so ago. Where are we as far as long-acting antiretrovirals are concerned? Clearly, we're talking about them for prevention and treatment. And we've got to identify those agents which can be combined, which are suitable for routine use. And we certainly need creative solutions going forward. Jonathan showed us the LATE-2, Cabo Tegravir with rilpivirine, where you had that induction period with oral, which was Cabo Tegravir, abacavir 3TC. Rilpivirine was then added in at week -4, and then people switched either to an 8-week or a 4-week injectable regimen versus the oral form. And what we had then was the effective maintenance with the injectable. But the pharmacokinetics are actually quite interesting in LATE-2, because what you want to know is what sort of drug levels do you maintain over that period after you've given your injections. And so you had a Q4-week injection for Cabo Tegravir, Q8-week injection for Cabo Tegravir, and the concentrations were pretty much above the protein-adjusted IC90, which is way down here, and commensurate with the oral levels achieved after oral administration of the drug. And here you see rilpivirine, and you get the Q4-week and the Q8-week. This is the protein-adjusted IC90, and the kind of levels that you get from the oral administration of rilpivirine. But in LATE-2, there was a couple of subjects who failed. This is one subject, subject 551, a week 48 protocol-defined virological failure with the Q8-week dosing. And in this patient with the Cabo Tegravir concentrations, you can see these low concentrations compared to the median values in the group as a whole. So this patient had something potentially wrong with the injection maybe, and the concentrations were very low in that early period, below what you were certainly seeing in the group. And same with rilpivirine. Rilpivirine concentrations were also low as well. So, you know, getting the injection right is going to be vital if we're going to have injectables. And then in another study that was done at Chelsea and Westminster with rilpivirine alone as prevention, there was one patient who was in the arm which had 300 milligrams of rilpivirine, and rilpivirine concentration shown in the blue here declining. You can see the declining rilpivirine concentration. And these were healthy volunteers. And here was a reported exposure with an HIV-positive partner. And you can see the viral load rising. And this patient got a K101E mutation. So drug concentrations were clearly important. And one of the worries with these long-acting is the tail. So you've got this tail, and this is the protein-adjusted IC90, so the tail is below that. And then in another study, which came from Ian McGowan, he showed this residual plasma rilpivirine concentration tail. So that somebody who has an injection and then doesn't return to the clinic for the next injection has the drug there. And we actually showed in this study that rilpivirine concentrations could be measured up to 2 years after a single injection. So you've got drug there for 2 years after that injection of rilpivirine. So clearly that's a worry, just hovering below the protein-adjusted IC90. So some of the key issues that we have to address: which drugs can be combined for long-acting? The injection volume, still a bit of a problem. 2 mils in each buttock is still quite a lot of injection site reactions. What to do about the missed doses, this long tail, low drug levels, management of adverse events that occur during an injection period. Are the DDIs going to be different? How are the regulatory authorities going to look at DDIs? How do we study DDIs with long-acting injections? And then we have this interesting compound, the EFDA, which is a novel nucleoside with a unique mechanism of action, which has been given to us at CROI, where you've got the half-life of 103 hours, and with a single oral dose of 10 milligrams, you get a viral load drop of nearly between 1.5 and 2 logs at 7 to 10 days. And in proof of concept, this drug is also being studied as a long-acting form with effective drug levels for more than 180 days in the rat and the non-human primate, and that's going to go forward. And then we have other formulations: tenofovir, alafenamide, TAF, the TAF implants, for example. This is a polypropylactane device. It's a little bit like contraceptive Norplant type of issue. And then we have those other devices here for delivering drug, which can be implanted, and so you can get drug which comes from that delivery as well. And that is something which is clearly of interest. My own unit in Liverpool, we have a big nanoformulations team, and the team in Liverpool are working with the USAID and PEPFAR and various other groups to study this whole idea of getting nano formulations of drugs. And then with the new antiretrovirals as well, which I haven't mentioned, we haven't really discussed in any great detail, I think we do have a future pipeline which is quite encouraging, and future paradigms which certainly are of interest. And I want to thank my colleagues. I have a lot of colleagues who I need to thank, but especially these two colleagues who are on the platform, who sit snoozing on the side while the others play the game. And so we're grateful to my colleagues as well. Thanks.